my great pleasure to introduce Dr. Joann Whalen. She's a professor and William Dawson Scholar, endowed research chair in the Department of Natural Resource Science at the Macdonald campus of McGill University. She's an adjunct professor at the Northeast Institute of Geography and Agroecology, Chinese Academy of Science. Joanne received her B.Sc. Agriculture from Dalhousie University, Master's from McGill University, and Ph.D. from Ohio State University. She's well known for her expertise in earthworm ecology and the contribution of soil biota to nutrient cycling and greenhouse gas fluxes from agroecosystems. Joanne has authored more than 170 peer-reviewed publications, graduated 43 master's and PhD students, and trained visiting scholars from 21 countries. She is the past president of the Canadian Society of Soil Science and a fellow of the Soil Science Society of America. Joanne is the chief editor for Soil Biology and Biochemistry and the subject editor for Applied Energy. Joanne also has collaborated with Andy and myself on agroforestry projects for many, many years. So she's not a stranger. She has come here. She has been an examiner in the Department of Land Resource Science and School of Environmental Science. So it's a great pleasure, Joanne. Thank you so much for coming all the way from Montreal. And without any ado, I will open the floor to you.
Well, thank you very much, Naresh, for that kind introduction, and thank you all for coming. And those that are just coming in, please, there's some chairs. It's safe up near the front of the room. I'm not going to leap out and make you do something. So please come in, filter in, and join as we'll talk for the next approximately 40 minutes or so about soil health. I'll give you some perspectives, and then in the end we'll talk about some opportunities and challenges that lie ahead. The big challenge, the big challenge of why we're all in the field of agriculture and why we're interested in agriculture is because we want to grow food and we want to ensure the security of food for the growing global population. Because the global population is large and it's not going to get any smaller, it is assumed that China's population might level off at some point in time, but India's population is projected to keep growing. And so it will be like this. The future climate that we're going to see is warming. So here are some projections for the temperature rise, and you can see that it's going up. The rainfall in the future will be more erratic. Sometimes it will be more intense. Sometimes it will be drier. Sometimes it will be wetter. We can't predict exactly, but we know that it will be more difficult, especially for crops, which are subject to stress and which don't do well under warmer climate conditions and with more erratic rainfall, because they're subject to all sorts of stresses: light, heat, cold, drought, salinity, heavy metals. But fortunately, plants are responsive to these stresses and they have mechanisms and ways to deal with them. So if you give plants a stress, they will respond to osmotic or oxidative stresses by producing stress signal sensors and transcription factors that activate stress genes. We still don't want to put our plants under stress, because the more stress that plants have, the less production that they can produce. So our sustainable agroecosystems of the future have got to be able to defend our crops against these stressors that they're going to be faced with, not only those abiotic stresses of the climate, but also those abiotic stresses are going to have an impact on biotic stresses. Pests may become more prevalent. Pest outbreaks and diseases may become more difficult. And we have another perspective to consider, is that we need to consider how pesticides that we have traditionally relied on may not be our best alternative in the future. In addition, our agroecosystems of the future need to conserve scarce resources. So they need to be conservative of water, nutrients, and energy, because not only will they be providing more food in the future, but they're going to be called on to provide more other things like fiber and fuels in the future. These diagrams give you some indication of the rising demand globally for biodiesel and bioethanol. Even we've heard about airplanes flying on biojet fuel. Actually, I'm part of a project, and we did a project in which biojet fuel was injected into the fuel system at Pearson International Airport back in December. It was at a very busy time of year. All that biojet fuel has been consumed by aircraft that were flying at that time. But it is one of the demands that's going to be made on agricultural systems is to provide more biomass, more residues, more materials. It's not only agricultural residues and materials, but it might be purpose-grown trees like short rotation willows, such as what the group of Naresh Srinivasan and Andy Gordon have worked on for many years. So thinking about all these demands, fuel, food, fiber, and the desire to preserve what we have for the future generations, we have to set multiple goals for ourselves. We have food security and quality goals, environmental goals, societal and cultural goals, as well as research and innovation goals. My topic today is soil health. And it is a concept that reflects our perceptions of how the soil is functioning. And so it's kind of a barometer or indicator of what's going on in the soil. Soil health is often equated to a biological function or measure in the soil, to the diversity and activity of the soil microbiome, which, as you know, is extremely diverse. So how many organisms are in that little teaspoon full of soil? Well, it's supposed that in a single teaspoon of soil, which is about 1 gram of soil, you might have 1 billion bacteria, several yards of fungal filaments, several thousand protozoa, and scores of nematodes. And if you looked in that soil, and you looked at it really closely under a scanning electron microscope, this is what you'd see. And what you may not realize is what you're seeing there is life. Because what we're seeing there is you're seeing some small bacterial colonies on the surface of the soil. And those are the food source for little protists that live in the soil. So they're driving around all the time, and they're looking to eat something, and there's their food source. And then, of course, there's fungal hyphae. All the sort of whitish filamentous material is the hyphae of the fungus. And those not only are in the soil because it's a great habitat, but they're also there to provide a food supply for fungal-feeding nematodes, which also live in the soil. So there's a vast array of biota that all live in the soil. And the soil microbiome is extraordinarily diverse. This, of course, is the phylogeny of the tree of life as we commonly know it. So what's all the blue stuff? Any ideas? Bacteria, right? So all the blue is the bacteria. That's the diversity of bacteria in the world. So most of the diversity in the world is bacteria. And then, of course, there is diversity in other protists like the archaea. And so they're in the green. And then all the red stuff there is all the eukaryotes. And we're a little tiny slice in there with the animals. So we're totally, totally insignificant in relationship to all the diversity that lives in the world. And so there is so much diversity and so many functions, and we're learning every day and all the time more and more about this. We can look in various places in the soil to explore that diversity. And so a very typical place that we would look is we'd look around the roots, because we know that there's organisms that live around plant roots. They either nodulate and produce nodules such as rhizobium, or they live around the surface of the root and all of that. Now they must have some importance, and they must be useful to the plant. They're not just freeloaders. We'll just go through a couple of examples. So this is the ask you questions part of the presentation. Does everybody know what this is? Right, we all know this crop. And here's another question. Maybe those of you in agriculture know the answer to this question. How much nitrogen does it need for its optimal growth? Actually, I don't know the recommendation for Ontario. What's the recommendation?
250 kilograms.
About 250?
Yeah.
Do you actually put that much on?
Yes.
Really?
Yeah.
Well, if you want to grow around 250 bushel corn, you're gonna need a great deal of nitrogen fertilizer, right? Because that's how much you need to grow the crop. And I'm just going with I'm being a little conservative here about a recommendation. The fertilizer recommendation, what's written in the book, might be about 150 to 200 kilograms of nitrogen per hectare. Actually, in the Quebec book, we're a little lower than that. We say 120 to 170. But if we want to be realistic about what people really do, we put on a great deal of fertilizer to support our corn crop, which we all know. Now, what's this crop? Sugar cane. So it's another grass crop, right? That's another grass crop. And how much nitrogen do you think it requires? More or less than corn? Okay, raise your hand if you think more. Nobody's raising. Okay, you think less? Okay, you think some people think less. Well, actually, if we think about what it really needs, it requires more. Look, to get a big, nice, healthy sugarcane crop, look how tall it is. Look at those little people beside it. They look like midgets. And the crop is big and it's tall because it actually is taking up over 300 kilograms per hectare, more than your corn, to produce 100 tons of cane. But the fertilizer recommendation for sugarcane is not very much. Interesting. It's a big crop. It needs nutrients to support it. But we have a small fertilizer recommendation. Why? It's endophytes, endophytic nitrogen-fixing bacteria, specifically Acetobacterium diazotrophius. And so this, I'm going to just read you a little quote that was from a paper. It was a research group out of Carleton that wrote this paper in plant physiology about 24 years ago. And so they wrote about this organism. This is an endophyte. So recently, a new microaerobic nitrogen-fixing bacterium was isolated in large numbers from sugarcane stems, roots, leaves, and rhizosphere soil from sites in Brazil, Australia, and Mexico. And now, I love the understatement that scientists always write in their paper. This bacterium, Acetobacter diazotrophius, has most unusual growth requirements. It shows optimal growth with 10% sucrose and pH 5.5. It will grow in medium with sucrose concentrations up to 30% and rapidly acidifies its surrounding by the formation of acetic acid. So it's an endophyte that lives all through the whole entire plant. And the reason that most sugarcane plantations do not need nitrogen fertilizer is because they have this organism and others, nitrogen-fixing bacteria, that will inhabit their entire body and will do the nitrogen fixation for them. And a lot of the reason that we can rely on them is because sugarcane breeding programs traditionally did not put much nitrogen fertilizer in, and so thus created a cultivar that was always very hospitable to support the endophytes that would naturally grow in it. We have to adjust ourselves to this thinking of the plant as being associated with microorganisms, and we need to update our thinking because even when I wrote some things and drew some pictures for a book that I wrote a few years ago called Soil Ecology and Management, this was my picture of the root and the rhizosphere. It looks very sterile. It looks like I'm just looking at the plant parts alone. So I have the root with some cells in it. It has root hairs. I talk about root exudates, etc. That looks like a very sterile thing. The plant root all by itself creates its own life, and it's kind of independent from the soil. In fact, the better way to look at this thing is that the plant root is surrounded by and has associations with all types of microorganisms. So the concept that we should consider is that plants are just sitting there and passively doing things or trying to exert their own control on the environment, but plants modify their environment and they build their own microbial community. So have you all heard of the soil microbiome or the plant microbiome concept? Right, everybody's heard of that. So let's keep thinking about that because that's important for how we develop our sustainable agroecosystem. The soil microbiome must be helping the crop, and it must be able to help it to resist some stresses. And of course, when we have this nice association together, then we can think of the agroecosystem as being more resilient because it will be able to withstand and recover from stresses that it encounters. We put together a vision recently that is a paper that Chen-Wei, Boloma, and myself wrote that we have in Advances in Agronomy, in which we discussed how plants, and we were taking canola as a case study here, will respond to stresses. The stress in this case will be the heat and drought stress that causes a response of the plant. First, the plant senses it. Where does the plant sense the stress? It senses it in its roots. The roots are the organ that first senses stress. So the root area and the root zone is extraordinarily important because that's where the first sensing of stress occurs in the plant. And then once the plant has sensed the stress, it may either choose to escape from the stress by dying or propagating and then dying, or it may choose to tolerate the stress. So a lot of plants have capacity to do that because they can do various things at a cellular level, like reactive oxygen species production and scavenging, change the fluidity of the membranes, etc. They can do various things in the transduction of various signals in the body through calcium signaling, post-transcriptional regulation, etc., which induce particular types of genes and transcription factors. But the most important thing about this whole thing is that they will want to change something about the root architecture. And the thing we must always remember about plants is that they're extremely plastic in their growth. They have this, always the G times E interaction talks about the phenology or the phenologic or phenotypic changes that a plant can do. And you know that genetically identical plants can grow quite differently given their environmental conditions. And the most important thing about this is to think about how the root system and how the root architecture can affect the plant's survival ability. And we should be able to use this knowledge to actually select cultivars and choose them in our breeding programs. We need to think about the root system because the root system is the interface at which all communication occurs with the plants and the microorganisms that live in the soil. So we see that the plant root is able to produce various chemical signals that it sends out to its partners. And through those signals, it communicates with them. There are some usual types of organisms interacting with roots that you see there, but there's also some types of signals that you might not recognize exactly what is going on. I will show a little more interesting picture to help you understand the whole concept. So the plant in this picture is saying, I produce root exudates that contain flavonoids, cytokinins, IA, volatile organic carbons, and other things. Can we be friends? These bacteria in the soil say, I also secrete these compounds, let's be friends. So they talk back and forth, and they decide to be friends with each other, which leads to a cascade of events and multiple interactions that occur here in which the plant and the microorganism take advantage of this understanding that they have to live together to produce things like, for instance, nodulating nodules in the plant with nitrogen fixation ability, or endophytes that invade and have nitrogen fixation ability. Some microorganisms that live in the environment will produce hormones like IAA that will help to change the root architecture. The cytokinins and the IAAs are important because you remember that they are promoting cell division and they're producing growing tips of the plants. And then, of course, there's also the AHLs, which are the N-acetyl homoserine lactones that are involved in quorum sensing, so they are communicating back to the organisms, and volatile organic carbon, which is a compound that also senses and sends signals back and forth to plants to say, something has happened, I'm under stress, I want you to know there's a stress in the area, be careful, the stress is there. This is well known for the above ground parts of plants. We know tree species do this all the time. If one tree gets eaten by insects, it produces volatile organic carbons to tell everyone around, be careful, there's insects in the area. They might come and eat you. Get ready. And they also do this below ground. So there's all this signaling going on, and the microorganisms can also act as a signaling network. And you can imagine this when we look at the mycorrhizal fungi. They fully colonize the roots of these little seedlings, and they're just extending their hyphal network everywhere. And so they see things in the environment before the plant ever can find them. Before it even gets close to the plant, the microorganisms, the fungal network here senses it and can give the message back to the plant and say, hey, guys, be careful, something's coming. And of course, this works out very well for plants because almost all plants are mycorrhizal. And so I remember having this conversation with a colleague who was a specialist on mycorrhizae, and I was talking about plants in my garden, and everything I said, she's like, oh, that's mycorrhizal. Oh, that's mycorrhizal. I said, I didn't know they all were. She said, almost all. They're almost all mycorrhizal. But of course, the one we know, the famous one, is the arbuscular mycorrhizal fungi. And we know it because it's important in agriculture. We've seen pictures of it. And we know that we can culture them, that we can isolate them. This one, Glomus inerates, that's a very famous one. And you might have even used it yourself. Has anyone ever used it? Okay, well, have you ever purchased this product? Have you ever used this product? Yes? No? Recognize this? Come on. You go to the plant store, the garden center, and you buy a tree. And they say, we'll give you one guarantee on that tree. But if you buy Mike's, which is like Mike's, mycorrhizal fungi, Mike's, right? It's a cute name. And it's cute, look, like M-Y-K-E, and the way they do it is very, very pleasing. So you want to buy this because you'll get a 3-year guarantee on your tree if you purchase this product. So you can purchase it. They have different containers. They have a green one for your tomatoes. They have this orange one for your trees. I don't know. They have different ones depending on what kind of plant you have. You can culture these things. You can isolate them. Have any of you ever used ProMix? ProMix, growing stuff in the greenhouse. ProMix is almost all of it's inoculated with Glomus inerates, okay? ProMix Plus might have a plus sign on it. It's got the mycorrhizal fungi in it. Okay, so they're going to do a bunch of stuff for your crops. They're going to be the early warning system to tell the plant, be careful, there's something out there. And in doing this, they don't just say, be careful, they actually induce the plant to do something. The plant, in response to this signal, will start to increase production of allelochemicals like terpenes, alkaloids, phenols, to make it more unpalatable, to make it more resistant. It also induces plant defense genes like phenylalanine ammonia lyase, which we also call PAL, also chitinase coding genes to break apart the cell walls of invading pathogenic fungi. And it also activates the jasmonate and salicylate acid signaling pathways in the plant. So that's the immune system. And the overall picture of what this looks like is something like this, in which you have a plant root and plant root system that will be colonized by these mycorrhizal fungi. And now it's not only protecting the plant because nothing else can get in and occupy cellular space because it's already fully occupied, but it also is the early detection system and sending the signal back to induce the systemic resistance. There's some other things there, but let's focus on the biocontrol parts. The induced systemic resistance is a cascade of hormonal effects, upregulation of IAA, upregulation of cytokinins, upregulation of ABA, abscisic acid, and downregulation of gibberellic acid and salicylic acid, to finally cause the upregulation of the jasmonic acid defense system. I'll show you one example to make it a little bit more concrete of how this might occur. And this is just one little example of many that exist in the world. And so this is a little story about a poor tomato plant. A tomato plant that was very susceptible and somehow got attacked by some pathogens. So the first thing that happened is Pseudomonas syringae came along and it managed to get itself into the stoma of this tomato plant. And so what happened is the plant recognized, oh my goodness, something bad is inside. And so what it did is it excreted a substance in the rhizosphere. It had a root exudate called L-malic acid that it started to exude out through its roots. It's like a call for help. I think I'm sick. In the soil, meanwhile, Bacillus subtilis recognizes the signal and says, got to help out, because they're a plant growth promoting rhizobacteria that's very helpful in this case. Bacillus subtilis starts growing like crazy. They start producing antibiotics. They start killing off everything around them and start a metabolic process in which everything else around them is suppressed, and they send signals back into the plant to upregulate hormonal pathways that end up to make the plant close all its stomates so no more infection can occur. So you can imagine how quickly this happens. Meanwhile, something else is going on. This poor plant is subject to all sorts of attack. And on the other side of its other leaf, Alternaria Solani is a fungal pathogen, and it's starting to colonize on the leaf. What this does is it makes another signal go down into the mycorrhizal fungi, and so they send some signals back to upregulate defense pathways, but meanwhile they're connected to neighboring plants, and they send a volatile organic compound over to those plants to say, be careful guys, Alternaria solani is around, so prepare yourselves and get ready. So this is the kind of thing that is happening in the soil at all times, and this is why biocontrol can be such a powerful tool in the whole toolbox of what we have to keep our plants healthy when they're encountering these type of stresses. I'll tell you a few other things about the mycorrhizal fungi. They're very important for water and nutrient acquisition, and it's illustrated so clearly every time we see those lone trees on a cliff, and you're wondering, how on earth do they grow on a rocky cliff? Or this one that is growing very happily. It looks like in a purely rock face of a cliff. And in many cases, the reason that a plant can live in an extremely hostile, arid, dry environment is because on the ends of all of its roots are mycorrhizae that descend down into tiny, minuscule cracks in the soil or in the rock, and they go much deeper and much more extensively than any root can reach because they can penetrate very, very small pores, and they can get water that is inaccessible to the plant itself. So more water and more nutrients is generally what we think about the mycorrhizal fungi, and that's illustrated in lots of different pot experiments in which we see organisms growing without the AMF treatment, and they're quite small, and then the AMF treatment just makes them much bigger, more happy, growing better. And we could think that it's because something to do with the nutrient status of the plant, but it might also have something to do with the soil stability. And so I'm going to talk about those two things. First, the nutrient aspect, and then the soil structure stability aspect of what fungi and other organisms do for the plants. So strigolactones are the signal compounds that will tell by which the plant will tell the mycorrhizae, I need you, help please. So this poor plant was going along, everything was good, you can see it growing, little root system, shoots very happy, but at some point it encounters a stress, phosphorus or nitrogen starvation, and so it responds by producing some strigolactones and calling out for help. I'm hungry, please help me. And so now what happens is that biosynthesis and exudation also changes the phenology of the root system. So we have some longer root hairs. We now have a higher density of the root hairs as well. The lateral roots, should have been mentioned here, more lateral roots or longer lateral roots. Also the trajectory, the positioning of lateral roots, because they can go this way, they can go like this, or they can go like this. They can change the angle of their roots depending on what kind of stress situation they find themselves in. Meanwhile, they inhibit the shoot branching because they're putting more resources below ground, and maybe they're producing some flavonoids to get the rhizobacteria helping out, nitrogen fixation, et cetera, get the arbuscular mycorrhizal fungi associated, helping them out. There's a whole series of responses, and it works at a physiological and cellular level, also at a genetic level. The exchanges between the plants and the mycorrhizae are affected by abiotic factors, agronomic practices, and things like the availability of nutrients in the soil. So this little diagram is showing the ways that actually in an association, both the plants and the fungi can, for instance, mineralize organic phosphorus. Both the plant and the fungi can produce organic acids or modify the soil pH to solubilize inorganic phosphorus, so they can work together. And then, of course, they have relationships in this direction in which they exchange materials. Phosphate goes from the mycorrhizae to the plant through high affinity transporter system. Plants give carbon, but they can regulate the amount of carbon they give to the mycorrhizae. So all of these things are in a very tight regulation with lots of feedbacks, and it's very interesting. Plants, however, are highly, highly dependent on mycorrhizae, as I said, and this was some results of a bit of a literature survey that Anna Albin and I did, and we looked at 3 crop families, the Solanaceae, the Fabaceae, and the Alliaceae, and we found that mycorrhizal dependency, meaning the proportion of phosphorus that was obtained for the crop that depended on their mycorrhizal symbiont was anywhere from about 40% to more than 90%. So most of the phosphorus in a lot of plants comes through their mycorrhizal partner. Next, we move on to soil structure. No plant is going to grow in a compacted soil. Every plant needs to have some space to put its roots. So we need to have an appropriate soil structure, which we evaluate by maybe digging up the soil and looking at it. But let's go down to a very microscopic scale and see how those soil microorganisms are helping us out in this regard. You know, all soil surfaces in the soil are coated with soil bacteria. If you don't believe me, go to this website. This is a very nice one, actually. It's hosted by the University of Bremen in Germany, and they have a tremendous resource with a lot of scanning electron microscopy of soil surfaces. So I'm just going to share with you a few pictures about this. Here's also the ubiquity of bacteria is pretty phenomenal, and this shows a fungal hyphae that's just totally colonized by soil bacteria. So don't think of them as like living separately or having separate lives. They all live together. It's just like you. Do you think you're really you, uniquely you? You're about 50% bacteria. So just don't think you're like only you and only you makes all decisions. The bacteria living in your body make a lot of decisions, and they help you out with many things, and you never even know it. Just hopefully they don't make you like think wrong things, or you could always blame it on the bacteria. Why did you do that? Oh, it's the bacteria. They made me do it. Here's what some of the bacteria in the soil are doing. They're producing mucilages, glue-like substances that help them to stick onto soil surfaces, and that helps them grow into biofilms and grow together. But it also helps hold the soil together. And here we see clay minerals being held together by microbial secretions, particularly extracellular polysaccharides. Other things that will hold soil particles together is actually the dead cell remnants of dead microorganisms, which we call necromass. So there's some extracellular polysaccharides that soil bacteria produced and that are actually binding clay's surfaces together. And then if we think about soil structure as being aggregates all held together, and these are just clay surfaces, but a real aggregate is held together, and it's often encapsulated and enmeshed in a fungal hyphae that grow all around the whole surface of it. So it's a very complex thing. So the organisms live in the soil, but they also make houses in the soil for themselves. And they also change their house every so often. They rearrange it, and they make new ones, depending on how long the compounds they've produced persist. Okay. Any other indicators of soil health? I've spent all this time talking about microorganisms as if they're most important thing in the world. They're the most important thing, but we mustn't forget that they live with other organisms. They live as part of a soil food web. And so the soil food web is held together because the soil food web depends on transfer of energy from the first trophic level all the way up to the fourth or fifth trophic levels and higher. So that's how a soil food web works, is that some organisms colonize some substrates, and then part of the energy and nutrients contained in them is moved up to the next trophic class as it consumes the lower trophic levels. And of course, we have microfaunal predators that in their quest to get energy and sort of swimming around in the soil and eating smaller things, they're very messy eaters as well. So as they consume this material, they don't eat it all, and they can't metabolize all of it. Their nitrogen use efficiency is maybe 10% or so, because they're like us, they're not totally efficient of all their food. A certain portion has to be defecated, and so that releases organic compounds that then get mineralized, and it releases ammonium and nitrate. This predation is important to regulate the size of the microbial biomass and its activity. But they don't graze on everything in a uniform way because they have preferences. So here's some study that we did. This is Ting Lu and and our colleagues at Nanjing Agricultural University that we had this study with a feeding trial, and we had a variety of bacterial species, and we fed it to 2 soil-inhabiting nematode species that are bacterivores. That means they eat bacteria. And you can see that the number of nematodes that went to eat these bacteria species are not uniform. The Pseudomonas fluorescens and an E. coli OP50 strain were the more preferred type of food source. So we were curious about why they have preferences for certain types of bacteria, why they're more tasty or something. And actually what it was is that the nematodes were preferentially going after these organisms because they were having a higher water content. So this led us to an interesting conclusion that protists, even though they live in water film, must get a lot of their metabolically active water from their food, just the same as you and I do. I mean, you get a lot of water from food. You can drink liters of water if you wish, but a lot of water comes in with your food. And the other thing that they were seeming to go after was the protein content. So they were looking for some nitrogen. We also have the soil macrofauna, and they're another source of organisms that contribute to the nitrogen cycle. They do a lot of things like make the plants grow better because they speed up the decomposition process, and they mix the litter in with the soil to make topsoil. They increase the cycling and the amount of organic carbon, nitrogen, and polysaccharides in the soil. They increase the porosity, aeration, and drainage of the soil as well. You will have seen this structure in the field, yes? Yes? Everyone seen these? Middens? Yes. And we know that the earthworms did this because every so often you see an earthworm wandering around with something in its mouth. And you say, okay, that's how it did it. But actually, it's at night they do this. They go out at night, they rummage around, they gather some things, they bring them back, they pile them up. And we figure that actually what this is it's a pre-decomposition type of arrangement, in that they're sort of composting the food before they're going to eat it. Because even the earthworm has a big piece of something in its mouth. It can't really eat that. It's too big, right? So it has to decompose, and they'll eat it afterwards. And then, of course, what comes in must go out. So the other end of the equation is the casts that come out as a fecal deposit. And so we also, in our research group, look at earthworms. And so, of course, I must show you some results of earthworm abundance in corn, soybean agroecosystems in Quebec. And it's really quite remarkable about how earthworm populations can vary during the year. So sometimes we have upward of, say, 500 individuals per square meter. And of course, in the drier times of the year, around July, the end of July, generally the numbers are much lower. And also, as we get farther on in the growing season and we get closer to winter, the populations tend to go down as well. But they're very much dependent on how much residue is there. So our corn system that had high residue because it was harvested for grain had more earthworms, and the corn system that had the low residue because it was harvested for silage had the ability to support fewer earthworms. And so the earthworm populations consumed a lot of material, and we can estimate, using some techniques, the amount of nutrients that they're recycling. And so their recycling activities can produce about somewhere between 22 to over 100 kilograms of nitrogen per hectare per year. This is good news for all those cornfields. Maybe some of the nitrogen supply for those cornfields comes from the earthworm population. And if we want to support this large earthworm population that can help us recycle some nutrients, we have to provide them with food, because their abundance is related to the food supply. And so here's just a very simple graph showing the amount of surface residue on the x-axis and the earthworm numbers on the y-axis, and you can see the correlation is there. So more food, more earthworms. Makes sense. If I brought food into one section of this room, I think you would all migrate there. Your abundance over there would increase very dramatically. If I didn't bring any food in, you'll stay well distributed where you are. You might actually leave to go find food. So that's what we find their abundance related to, food. Not necessarily indicators of residue quality, just food in general seem to be the most important. Okay, so now we come to the perspectives part. What do we do in the future? Well, I think we need to ask ourselves the question, what do you need or what do you want the soil microbiome to do for you? Do you want it to help you with plant protection? Do you need it to help you get more water? Do you want it to help you with better nutrient acquisition or better nutrient use efficiency? Do you need the microbiome to help you improve the soil structure? No matter what you choose here, there are ways to make the microbiome more useful to you. And one of the first ways that I think that we can do this is to, first of all, select some crop cultivars that are compatible with the microorganisms that live in the field. And I think that plant breeding programs, as we continue forward, should not only consider root architecture, but should consider root architecture in how those roots are going to interact with microbes. Just a little picture to show you and illustrate this point that I'm trying to make. These are two seeds that have been colonized by fungal hyphae, and you can see that the colonization is unequal. It's because the signaling and the compatibility is not uniform in all plants. So if we think that all plants are the same in terms of their ability to connect with microbes and to speak with them and to interact with them in a beneficial way, it's quite not true. So we must be sensitive to that moving forward. The other thing is that we can continue to work on efforts to develop beneficial arbuscular mycorrhizal fungi, or really any type of beneficial microorganism for specific crops. And all that relies upon is some type of work in which we would isolate inoculum from roots, and then we can either go forward with a single species that we can grow up and have as a single culture, or as multiple species, such as these commercial products here. So you can get those and you can purchase them and then you can apply them to specific crops. But for those of you working in a field condition, just realize that there's a tremendous diversity already in the field. So it might be, maybe this is used more in a greenhouse setting or in a controlled growth environment, whereas our other idea about breeding programs is very much important for our field setting and making sure that we're compatible with plants and microbes in the field. And finally, I think that we'd like to make sure that we always have enough food for the soil food web so we can optimize their performance. And so by food, I generally mean organic material coming into the system, so manures, crop residues, cover crops. Keeping the soil vegetated at all times is very helpful in this regard. And also making sure we have compatible host crops in our crop rotations so that we don't go for long periods of time in which there isn't a plant that might be associated with a particular microorganism that we'd like to keep supporting. We also can consider how to manage the soil to keep favoring the growth of the soil microbiome. We know from the phosphorus work that high phosphorus levels will kind of impede the mycorrhizal fungi because the plant has no need for them if there's lots of phosphorus. Same thing with nitrogen fixation. If you have lots of nitrogen already in the field, you will inhibit nitrogen fixation because the plant won't invest in that. Here's one that I put, but with a question mark, because I'm not sure that this is the right answer. Some people have suggested to me that less agrochemical use, and here I'm talking specifically fungicides, might be important. I'm not totally convinced about that because even in our own research, we've tried to sometimes suppress certain organisms in the soil, and with a fungicide or a pesticide, you can do it, but for very limited periods of time. Biological survival is a very, very strong instinct, and it's really difficult to completely kill off or eradicate an organism living in the soil. It's, had all these bacteria and fungi have had much longer evolutionary time than us to adapt themselves to the environment, and so they're pretty resistant. And so I just put that as a question mark, but I'm not convinced that it's totally true. So it brings me to the end of the talk, and with that, I will just say thank you, and I will welcome any questions.
Please feel free to ask questions.
Yes, well, first of all, just a wonderful presentation. Very much enjoyed it. Is there evidence, you talked about VOCs given off by plants when they come under stress.
Yes.
Is there evidence, particularly in horticulture, that a plant grown under stress will give off more toxins that become carcinogenic? harmful to human consumption?
That's an interesting question, and to be honest with you, I haven't come across anything like that. I think most of the time when we think about plants growing under stress, we're concerned about the stress perhaps impeding their yield. But in terms of the buildup of some types of compounds in those plants, I'm not aware of that. I don't know if you'd heard any examples along those lines.
No, I'm curious.
Yeah, I actually don't know about that. I don't know if anyone else has any reflections on that particular point. We'll have to keep looking into it.
You didn't really talk about physical disturbance and tillage
Yes.
impacts. So can you comment on, we're trying to, reduce tillage
Of course.
and what its impacts is on the fungi and that?
Yeah, and so, that's a topic that I find a little bit controversial because a lot of our tillage activities are being done at quite a large scale, at a person scale, and organisms live at a rather smaller scale. And so very often what we do at a big scale could have a temporary impact on those organisms, but I haven't seen evidence that it would have a persistent impact on them. So I've seen that even with intensive tillage, that there's some disruption of, say, fungal hyphae, etc., but that is for short periods of time after the event has happened. After the soil stops being tilled and it's back to a non-disturbed state for a period of time, they will regrow. It just takes them some time.
So what's that time frame? Like, what do you mean by short-term months?
I think what has happened in some of the literature is that people will till and then within a certain number of days, like within a few weeks after the tillage, they'll measure an effect. But if you went, say, 1 or 2 months later, you might not see that effect. And I will give you an analogy. In earthworms, which I'm more familiar with, if you till, you can have immediately a reduction of, say, 90% of the earthworm numbers can be reduced because you'll start cutting them up and all of this with a rototiller, etc. But if you go back, say, 2 months later, the earthworm numbers are back to normal. So the populations can go up and down. And it's very difficult to imagine a tillage activity that can eradicate any type of organism living in the soil.
Just to make sure I have it right, there's this feedback system in response to pathogens. You're
Yes.
saying that so a pathogen colonizes a plant, and
Right.
then there's some exudation that comes through the roots.
Yes.
And then the soil microbiota then produces a signal in turn, which then causes the plant to produce something to respond to the pathogen.
Exactly.
It seems like a very circuitous. The plant has a capacity to respond to the pathogen, but it has to go through this circuitous, like, let's tell the microbes that we're under attack, and then the microbes will tell us to defend ourselves.
It's, if I can explain it, the plants do have ability to respond, but sometimes it overwhelms them. It's like even ourselves. We sometimes get sick, and sometimes we get really sick. If you have seen this type of organism before, or you've been exposed, you have a priming effect. So that's actually what the plant growth-promoting rhizobacteria are supposed to do, is they're supposed to give a priming effect. So they give like an extra boost to the immune response. If they were absent, the plant has to rely on its own defenses. So it can rely on its own defenses to a certain extent, but it actually has a better chance of defending if it is also being helped by the rhizobium, or by the rhizobacteria that are living in its root zone.
And are the rhizobacteria directly helping, or are they triggering the plant just to bump up its own response?
So they are producing hormones that then are transmitted back to the plant, and that stimulates a genetic cascade, and so an immune response in the plant. And what they're getting from this is they're getting a healthy plant, which provides them with food, which provides them with root exudates, not only in the signal molecules, but all of the 20% or 30% of the photosynthates that go down through the root zone. So it's in their interest to have a healthy plant, because all the time as the plant is growing and it's photosynthesizing, it is putting photosynthates into its leaves and into its own biomass, but it's also exuding, it's losing 20 to 30% of it through the roots. It just goes out through the root caps, through the root compounds, et cetera. So it's in the interest of the bacteria to keep the plant nice and healthy. That's why it does the priming
It's
effect.
in the plant's interest to keep itself.
Well, of course, yeah, of course.
I have a quick, I'm sorry. Go ahead, yeah.
In reference to your chemical question mark, just as a casual observation, blueberry growers in Prince Edward Island were spraying a lot of fungicides around against mummyberry and various other fungal diseases on lovage blueberries. And around the edges of the field, all the conifer trees turned red.
Oh.
And we assumed that it was the fungicides acting on the mycorrhizal, which were associated with the conifers, that caused the conifers then to go red and die. Now, you're probably correct, that would all rectify itself within a few years. But it is just, an example. We've been working a lot with Clonistachys rosea as a plant amendment, and that has a lot of similar effects to the sorts of things that you described, with bringing on additional plant health, additional plant vigor, and additional resistance to infection by various kinds of plant pathogens, notably Sclerotinia, Malarinia, and Fusarium, Pythium. All of these sorts of things seem to be suppressed by the presence of Clonistachys. Now, we don't know what the mechanism is, so it'd be very, very interesting to discuss further what might be going on there.
Right. Well, thanks very much for the observation about the fungicide effects, because that's, where I guess my question mark was. I hadn't run across, experimental evidence, but that observation is very useful.
It's just anecdotal, I'm afraid.
Yes, and also I'm not familiar with your organism. What's its name again?
Clonistachys rosea.
Clonistachys.
Cleocladium.
Okay. But the Clonistachys, that's its impact on all of these vast array of these fungal pathogens is extraordinarily promising. And so I think you've got something quite interesting there. It will be very, very neat to know how exactly it's manifesting these effects, whether it is through physical barriers to the infection of the pathogens, or whether it is through genetic means.
We just don't know, but they're certainly under cultivation by commercial enterprises. So, we know there's the private sector is looking closely at them.
Very good, very good.
I think we have to stop there because I think the other group is outside the door. So as a token of appreciation, Joanne, it's a little bit of honey from University of Guelph.
Thank you very much.
And a pen from our School of Environmental Science.
Thank you.